Hello everyone, and thank you for tuning in to Science on a Sunday. I'm Gita Yadav, and I'm a lecturer at the Department of Plant Sciences. I'm also holding a joint appointment at NIPGR New Delhi. Today I shall be talking to you about the phytochemical landscape, the dynamic mosaic of changing chemistry that surrounds us in space and time at all times, and how we're trying to decipher the silent language of plants. So plants have evolved some of the most advanced forms of communication on the planet, and they communicate with each other as well as with the rest of the biosphere through chemicals. Unpacking these chemical arsenals is not just about pushing and merging the boundaries of modern technology, but also about being able to understand and appreciate how this chemistry is pivotal to human well-being. Phytochemicals are all around you, and as Mark Hunter has put it perfectly, all of life, as you know it, is complex chemistry in a semi-permeable bag. If you've been to the south of France, you'd smell the lavender before you even see it. If you've gone trekking into the Himalayas, you're familiar with the bluish tinge that transforms the forest as soon as you enter or cross the foothills. The scent of jasmine blossoms, the yellow of sunflowers, the flavor of cinnamon, all of these and much more that you can't see or fathom are part of a variable code, a chemical code of plants. Variability being the keyword. There are more than 35,000 unique biochemicals produced by plants that are not required for their growth or development. So what are these guys doing? Isoprenoids released as volatiles constitute the most diverse group of these phytochemicals, and this is the group that we're investigating. The full complement of isoprenoids in a given species is known as its terpenome. Billions of tons of volatile terpenes are emitted annually by the plant kingdom on this planet, mediating specialized adaptations like defense, communication. Terpenes have also been known for several industrial uses like biofuels, feedstocks, fragrance, flavor, medicinal chemistry, and so on. Coming back to the variability in plant chemistry, it is not just in structure of the emitted compounds, but also in their function. And this variability is evident at all levels, as you can see in this image. Within and between species, within and between individuals, between tissues, developmental stages, and so on. I've divided this talk into three parts. The first part is about how we capture and measure the variability in emission profiles using a combination of real-time experiments and literature mining. The second part of today's talk is about how we study the variability by combining machine learning, genomics, and complex network approaches in the lab. And I'll be giving a couple of examples from the work we've been doing on invasive species. In the final section, I shall discuss the untapped potential in our collections at the Cambridge University Herbarium with the help of genetics and genomics combined with phytochemistry. So, how and where do we get the emission data? Conventionally, we identify small molecular weight compounds emitted as volatiles from plant headspace by performing a steam distillation followed by gas chromatography and mass spec. For larger molecular weight compounds, we perform an HPLC and so on. These processes have been improved and improvised over the years in several ways to capture headspace in the field using dynamic pumps, PMEs, and portable GC machines like the Xenos. Even under the most conducive conditions, there is no way for me, or for anyone else for that matter, to experimentally analyze or measure the chemical fingerprint of each and every plant species on Earth. But it is possible to do the analysis on a large enough scale which allows you to draw statistically significant patterns. And our premise here is that much of the information that we seek already exists in published literature. This story is about how we extracted and compiled plant emission data from literature. The work began 15 years ago when we searched open resources like the PubMed and created the Essential Oil database. This database is freely available at the link given here and presently contains over 150,000 essential oil records spanning 100 years of published papers, representing about 8,000 compounds, 2,000 species from 92 plant taxonomic families, and one of the greatest advantages of this database is that it has contextual data, which means not just the emitted compounds, but also information about add-on information about the emission, such as the time that the emission took place, the part of the plant from which the emission was captured, the source of collection, the geographical location where the collection was made, also prevailing conditions like stress and other things, if available, and so on. One of the downsides is that the SOLDB, or the Essential Oil Database, merely represents a tiny fraction of what we could potentially find in the full scholarly corpus of published data if only we had greater access. We are convinced that we will be able to greatly increase this database and expand the scope of analysis that we can do with a search of the full scholarly corpus. And the good news is that we do have a ray of light in this direction, and as I'm going to be explaining in the next slide. With the help of American technologist Carl Malamud, we have gained access to a gigantic store of article metadata extracted from 73 million journal articles dating all the way from 1847 up to the present day. And this has happened very recently. The big question whenever you start to look at such large corpuses of published literature is about not wishing to breach copyright. And in order to avoid doing so, we intend to build code that would crawl over the metadata, scanning through all of the world's scientific literature and pull out insights without actually reading the text. And this is not an impossible thing. It's very doable. We just need to write a couple of lines of code that would extract or retrieve information just the way we want it about plants and their chemicals indeed. And what we need is a set of dictionaries or lists of words that are important that would be searched or scanned by the code. So these are currently being built with the help of the CEV Open project. CEV Open is a collaboration that we have with Content Mine Cambridge and Very Clear USA. It represents a virtual citizen science community which is growing by the day, creating open modular knowledge and tools such as softwares and dictionaries to read open published scientific literature, also to make the literature semantic. So what we do is we crawl the metadata for three things. A, any plant species and their plant parts. So we've already got a full list of known plants that we term as a dictionary. B, any volatile compound. Again, we have large lists of dictionaries of thousands of compounds in different categories among the terpenes, non-terpenes, and so on. And C, where this was reported. So in this section, we would have information on location, collection time, anything else. Like I said, the SOLDB is contextual, and we wish to keep it so. So any kind of contextual information that might be important. Now, those dictionaries have got to be very carefully built in order to extract just the right kind of information. And the other issue, of course, with collating large volumes of chemical data involves complexities that are associated with data disambiguation and normalization that need careful attention. This is one example where we've applied it to plants and their medicinal oils. Emission data analysis. This is the second part of my talk. And in this section, I'm going to be giving a short snippet of what we're doing with the enormous amounts of data that we have collected and continue to collate. The focus of this section is going to be on invasive species. One of the major side effects of the seamless movement of plants and animals across the world that began in the Victorian era and continued further with globalization, ending very possibly only with the COVID lockdown that has been enforced upon all of us, was the introduction of invasive species or plant immigrants. With no natural predators to control them on new soil, one man's flower turned into another man's weed. Some of the most aggressive invasives can be introduced into a very small area and then escape out or explode out, competing for nutrients, light, pollinators, seed dispersers, basically crowding out other pre-existing species also sometimes called native congeners, and so can lead to species decline or even species extinctions. And biological invasions are one of the most important threats, very well understood to be one of the most important threats to biodiversity, and a global issue in ecosystem conservation. In India, particularly, where I work, the problem of alien plant invasion is quite severe. About 40% of the species in the Indian flora are alien, and of these, 25% are invasive. Some of the most disruptive invasive species have become only too familiar. The Japanese knotweed and the Rhododendron ponticum in Britain are very well known to most of us. If you've gone camping to the Lake District, for example, or along the Ravenglass railway tracks, you've got hills infested with Rhododendron ponticum. Similarly, we have invasives like Lantana camara, Ageratum conizoides, and Prosopis juliflora, many others in the Indian subcontinent. So what we have done is to collate as much information as we could from the essential oil database pertaining to invasive species, and the species for which we were able to find significant volumes of data both from invaded and native habitats are listed on this slide, and these were the ones that we investigated further. This work is an ongoing effort of curating thousands of chemical compounds captured as emissions of headspace volatiles from one of the world's most aggressive plant invaders, Lantana camara. Over the past 5 decades, several groups have tried to identify composition profiles of the BVOX produced by Lantana camara under varying conditions and contexts in both native and invaded habitats. What we have done is to compare foliar emissions from invasive populations in various invaded habitats across India that we've collated ourselves, as well with some of our collaborators. And then we compared these profiles with existing composition profiles of the same species from native and near native habitat, which for Lantana at least is South America. And these were available in the published records. With we only took those records for which we found as little variation as possible in the sample collection and profiling method, so as to have a reasonable comparative situation. Very recently, one of my Part 3 students in systems biology at Cambridge, Ewan Salter, constructed a neural network-based classifier to demonstrate the feasibility of being able to use plant emission profiles as unique species identifiers. And Ewan assessed the congruence between plant chemotaxonomies and phylogeny. What we found was that there is indeed evidence for the existence of multiple chemical variants in the invaded habitats of Lantana camara, one of the world's most aggressive invasive species, as I've told you already. We also find a strong signature for differences in both quantity as well as quality of emissions from native and invaded habitats, providing indirect but strong evidence for the novel weapons hypotheses of invasive species evolution and success. One of the biggest advantages of having geographical emission data, as we have done in the SOLDB, is that it can be mapped onto other kinds of data sets and information portals, such as A, the World Trade data, and B, species identification records which are available in the GBIF database. For example, we use the first report of observation of a given invasive as a proxy for the first appearance of that species in that region. And then we check the existence of connecting evidence to other native or invaded habitats or regions in the world among the records in the World Trade data around the same temporal window which we found the proxy information in GBIF. So this gives us a handle on when the species might have arrived in a new location. We did this correlation for the SOLDB records of Ageratum conizoides, and we were able to reconstruct the movement and invasion history for this major invader across the major continents, as shown over here in this map. The red arrows are our predictions of when we think the time that we think invasion occurred from one region to the other, and you will notice at least 5 different directions of invasion or routes of invasion after mapping this invasion history. This was further studied in the next slide, where we found additional support for phytochemical expansion. So in the previous slide, you noticed what I showed you was the comparison between World Trade data, the geographical location information in SOLDB, and the GBIF information of species records and observations. We then found further support for phytochemical expansion by statistical comparisons of the emission profiles themselves. So we took the complete emission profiles of Ageratum conizoides, individuals between the native habitat and the invaded habitat, spread across 4 major continents. So South America is your native habitat, everywhere else on the planet is invaded habitats, and this suggested very distinct biogeographical patterns of emissions, as you can see in the plots, in the box plots, as well as on the right side, the profiles where are the mean dendrograms of the profiles based on geographical region. This provides evidence again for the novel weapons hypothesis. So the next logical task that we had was to find if this difference that we have observed in the chemistry is a result of distinct biosynthetic pathways giving rise to the diversity of compounds, or are the same pathways being regulated differently potentially to produce the observed differences in chemistry? Or, most confoundingly, are these species able to create different products using the same basic set of enzymes and regulators? And that would be confounding, and as we find later, that might just be the case with these guys. At any cost, what this brings us to is the third part of today's talk, where we start looking at the genes and the proteins and the biochemical pathways that are responsible for producing the diversity or the variability in chemistry that we've observed. So the last part of my talk is about genomics and the herbarium. This is the CUBG that most of you are very well aware of, and recent years have witnessed a surge in genetic and genomic investigations across the world using samples taken from herbarium specimen, providing an additional window to research. If you just look at living plants, or let's say if you just take a look at the herbarium specimen on their own, even fossil information, what you get is a single snapshot in time. And that snapshot could potentially be dependent on preservation status or conditions at the time of collection. The principal advantage of genetic analysis is that you get an additional temporal window afforded by the length of time scales combined with the spatial breadth of the data in the herbarium. The CUBG Herbarium, the Cambridge University Botanic Garden Herbarium, offers an incredible resource for researchers. And if you heard Lauren Gardiner, the curator of our herbarium in the last Science Festival, you would be aware that we've got John Lindley's entire collections, as well as a huge treasure trove of type specimen from Darwin's Voyage of the Beagle. So what is our interest in the herbarium specimen? What are the genes that we want to look at among the specimen of the kinds of species that we have? As I said earlier, terpenes are some of the major components of phytochemical biosynthetic pathways, and elucidation of the product preferences for terpene synthesis, the enzymes that make these products, by correlating the genome to terpenome, would pave the way for rational design of novel terpene synthase enzymes, as well as give us a very interesting opportunity for synthetic biology and bioeconomy. So what we did was to scan over 50 fully sequenced plant genomes to identify terpene synthase genes and predict their substrate specificities to the major classes of terpene products like monoterpene synthases, sesquiterpene synthases, and diterpene synthases, and so on. So what we found was that some terpene synthases have the ability to make more than one very distinct compounds. As stated earlier, my student Yuvan Salter has also developed a neural network classifier using supervised and unsupervised machine learning on SOLDB data. We hope to further improve this classifier by including information layers that take into account each type of TPS enzyme and its action. And one of the major questions that we're asking is, are the invasive species terpenomes enriched for the kind of enzymes that we suspect are there in some of the 50 species that we have noticed? In the future, we'd like to bridge the glaring invasion genomic divide by combining the work that we've done with the genomes of 50 plant species, the terpene synthases, and by taking herbarium specimen and looking at the sequences of terpene synthases in these specimens across time and space, and to be able to see if there are specific single nucleotide polymorphisms in the terpene synthase sequences, and whether those single nucleotide polymorphisms might be responsible for the diversity or variability in the emission that we have observed. So we'd like to bridge the glaring invasion genomics divide, and we'd like to explore if invasive species terpenomes are enriched for unique TPS enzymes that can create multiple products from the same active site architecture. In the end, I would only like to reiterate that the dazzling variation in plant chemistry is a primary mediator of trophic interactions like herbivory, predation, invasion, parasitism, disease, you name it. And at the same time, such interactions feed right back to influence spatial and temporal variation of the chemistry that we see in plants all around us, that I spoke about in my first slide today. We are investigating the shifting spatial and temporal mosaic of plant chemistry within and between species, within and between populations, within and between geographical locations, with the hope to be able to identify patterns that will allow this largely scattered field to serve as the nexus between as the bridge between genomics and chemistry. I'm grateful to my collaborators, my students, friends, colleagues, funders, Suresh, Peter, Lauren, Carl, Emmanuel, and of course to everyone who cares to listen to Science on a Sunday. Thank you once again. Signing off now.